All right, so today we're going to go over osmoregulation. So here is a picture of sweat. Sweat is actually something we've discussed before when it comes to thermoregulation. Do you recall why sweating is important for thermal regulation? It actually came up last time when we looked at how your body maintains a constant body temperature. What's the role of sweating? Let's try to do. Anyone want to guess? Tries to, sorry? Tries to cool your body, is that what you said? Because I can't, for some reason your mic is all muffled up. Yeah, so it tries to cool you off, right? Now, the thing with, is that in the, remove heat from the body? Yes, very good. Thanks, Renee. That thing about that is, actually is a nice little tie-in to our next topic, which is on osmoregulation, which is your body trying to balance out water and stuff in the water, dissolved substances. And here's the idea. I mean, if you're losing water to cool yourself off, then there's going to be a point where maybe you're losing too much water, right? And then what happens in that case? Because you're sweating because you want to cool yourself off, but at the same time you don't want to lose too much water either, because that will lead to dehydration, right? So we're going to talk about how your body regulates water and dissolved substances. And we are going to focus on the kidneys, all right? That's what we're going to talk about primarily. But I do want to talk about, just in general, what is osmoregulation? So this is like trying to maintain balance of water and the dissolved substances in the water. And there's different ways, organisms do this. We are going to focus on mammals, okay? And we're going to look at how kidneys do this, because kidneys are going to be the star of the show when it comes to osmoregulation. Now, well, we need to think about a couple of things. One is you don't want to lose too much water, but sometimes you need to get rid of water because your body produces waste, right? And one of the ways we get rid of waste is by urinating out the toxic product. So here are 3 examples of waste products that are produced when we break down, certain things. So I'm going to focus on things with nitrogen in them. Okay. So we know that from your biochemistry, like proteins and nucleic acids, not your carbohydrates, not your lipids, contain nitrogen. So what happens when you break those things down? So when you break down amino acids and nucleic acids, you produce waste products with nitrogen in them. You can see that there are 3 here: ammonia, urea, and uric acid. Now, the difference between them is, you can see in this chart, that as you go from ammonia to uric acid, it's more expensive to produce the uric acid, according to this chart. So there's more energy to make it, but less water is required. So something that produces uric acid loses less water. On the flip side, something that produces ammonia, it's easier to make, but also requires more water to get rid of it. So if you're a terrestrial animal, if you live on land, water is an issue. Like, you don't want to lose too much water. That's a problem. So if you're a fish and you live in water, and you're surrounded by fresh water, does it really matter if you lose a lot of water? You can just, you can just drink more water. It's not a big deal. So what we tend to see is that in fish, so let me show you an example of fish. This is like a freshwater fish, okay? So a freshwater fish will produce ammonia, which means that they produce lots of urine, but that's not really a big deal, because they can just, take in the water because they're in an environment that is just full of water, right? So that's not a problem for them. But we're terrestrial animals, so we cannot produce ammonia because it takes a lot of water to get rid of it, and that would be a problem for us. So we produce urea. Urea, as you can see somewhere. Well, hold on, let's back up a little. We produce a little bit of everything, but mostly urea. And why mostly urea? Because it's kind of like that Goldilocks. It's not too hot, not too cold. It takes more energy than ammonia to make, not as much as uric acid. But you can see it takes less water to get rid of it. And that's important because the theme in this section will be conservation of water, right? And the valuable substances in the water. So, let's go back to this diagram of the freshwater fish. So, according to this diagram, freshwater fish will gain water because it's in an environment full of water, right? So it's not a problem. But it says it loses salt. So what's going on here? So we need to actually back up a little bit to all the way back to grade 10. In grade 10, I think in grade 10, you learned about osmosis and diffusion. Do you remember that topic? A long time ago? Osmosis and diffusion. So let me refresh your memory as to what that is. Okay. Let me just hold on. Let's see. Can you girls see this screen? Let me know.
Yes, sir.
Yeah. Okay. Good. So if you remember, because diffusion actually came up when we talked about making energy. If you remember, that came up when we looked at how ATP synthase works. So diffusion is when things move from a high concentration. Let's imagine this is the high concentration, this is the low concentration. And we have lots of stuff here on the left, if you remember, and very few on the right. And if they can fit through an opening, molecules will diffuse from high concentration to low concentration. That's just diffusion, right? Now, if you remember, water will also be affected also by solute concentration. So, and that's actually specifically referring to something called osmosis, which is the diffusion of water, okay? So osmosis is actually the diffusion of water. So if we look at what happens to water, if I have an environment like this, let's imagine that this is a beaker and there's some sort of a membrane in between, okay? And that membrane will allow water to go back and forth. So water can go through the membrane, okay? But there's a solute in the beaker. On one side, there is a solute. I'm going to draw it as these little red dots, like this. Okay. And on the other side, there's less. All right. Now, we talked about diffusion, which is things moving from a high concentration to a low concentration. Osmosis is specifically the diffusion of water. And one really important thing that affects it is the solute concentration. Okay. So what happens is water will travel to the higher concentration of solute, which in this case would be on the right side. So what would happen over time then is that on the left, because the water is moving to the right, you would get less water on the left, and you would get more water on the right, because the water has moved this way, because the solute concentration on the right is higher. There is some terminology that you might recall. If an area has a higher concentration of solute, that area is called hypertonic. And if the area has lower solute concentration is called hypotonic. And then when everything is balanced out, then that means the two sides are equal. So that would be isotonic. Okay, I don't know if do you recall this terminology from junior science, girls? I know it was a long time ago. Now, there's a reason why I'm going over this, because this is actually going to really help you understand how the kidneys work. If you understand diffusion, which is, again, things moving from a high concentration to a low concentration, like you saw with ATP synthase and how it works. And if you understand osmosis, which is water is going to want to move to the area that has more solute, then you're really going to understand how the kidneys work. It's really, really important. It's also going to help us understand what's going on this slide. It says that this fish is going to gain water by osmosis. And that's because, think of it this way. If this is a freshwater fish, that means it's living in an environment, it's freshwater, that is hypotonic. So it has low solute concentration. Now, the fish then, by definition, would be hypertonic, because if one side is hypotonic, then the other side would be hypertonic, which then explains why the water is going in. Because if the fish has more solutes inside than outside, water is going to go in. Now, if water goes in, the more water goes in, it's like, you know this, the more water you drink, the more water you have to get rid of, right? So the fish is accumulating a lot of water. It's also producing waste. It's producing ammonia, which it wants to get rid of. So, it's going to produce lots of urine, right? Now, if we look at the next slide, you'll see like the opposite. Here's the problem. The next slide, we have the fish is in a marine environment, which in other words means like the ocean or the sea. In that case, the fish is actually in an environment that is hypertonic, which means it has a very high solute concentration. Okay, which then by definition means that the fish is hypotonic. So, according to osmosis, water is going to leave the fish. Now, if it leaves the fish, that would mean then that the amount of urine produced, because now the fish has a problem, right? It's going to be losing water. So it's going to then adjust by losing less water through the urine, right? So that's one adjustment that we'll see in fish that live in a marine environment. The other problem is if it's losing water, where does it get the extra water from? Like, it's got to be able to replace the missing water. So it actually has to drink the salt water. Now, that's interesting because, see, we can't do that, right? If you were stuck in the middle of the ocean and you had no water, you would not be able to drink the ocean water. Does anyone know why you would not be able to drink the ocean water? What the problem would be?
It has too much sodium.
Yeah, it has too much salt. And actually, it would be the same as what happened in this beaker. Imagine this. Imagine that you drank the salt water, right? This would be your body, and this would be the ocean on the right. What happens if you drink ocean water is your body would actually lose water because the ocean water that you're drinking is saltier than the solution in your body. So it wouldn't actually hydrate you. It would actually dehydrate you. It would kill you in the end. So, you know, how do fish get away with doing that? Well, they have an adaptation. What they can do, this is what we can't do, is they can drink the salt water because they have the ability to get rid of the salt. We can't do that. So when we drink the ocean water, there's all that salt in it. That salt dehydrates us. But if we could filter out with salt. If we had a way to get rid of the salt. And, this is actually what some little island nations do. When, if you're living on an island, how do you get fresh water? Well, what they have to do is they have to remove the salt from the water to get fresh water. And fish have this adaptation. So they can do this. So they can drink the salt water because they can remove the salt. So here's a curious question. Mammals can't do this. So what's a whale? What is a whale, girls? Is it a fish or a mammal? What do you think? A whale is
Mammal.
yeah, it's a mammal. All right. So if mammals cannot drink ocean water, the question is, where do whales get their water from? Because whales are going to produce waste, just like all other animals. And we know that a lot of that waste gets removed through water. So then you're going to lose water. So how do they replace the water? How does that get replaced? Can't drink it. So what do? Does anyone know?
From what they eat.
Yeah, very good. It's from what they eat. Because what they eat has filtered out the salt, right? They filtered out, those things have been able to filter out the salt. So they get their water from eating things because they can't drink the water. So, if we understand what's going on with these two scenarios, it's going to really help us when we talk about the kidneys, which is actually what we're going to focus on. So we're going to turn our attention to how this works, okay? So we said that if you're living on land, the term for that is you're terrestrial, all right? Now, the problem with a terrestrial organism is because it lives on land, it has to figure out a way to conserve water, okay? That's going to be a major issue, conserving water. If you're a freshwater fish, you don't have to worry about that. But, if you live on land, you do. So we are going to focus on how the kidneys work. All right. So here's a nice, beautiful picture of the kidneys attached to, you can see the bladder. The bladder. All right. Right there. These structures are called the ureters. So what happens is your kidneys will filter out your blood, right? And they're trying to do two things: trying to get rid of waste, but at the same time trying to keep as much water as possible. Can't keep all the water because you need some water to get rid of the waste, right? Because your urine is actually mostly water. So whatever they're trying to get rid of is going to go down through these ureters, okay, into the bladder, and then you're gonna pee it out, right? So here is you're seeing a picture of a horse. This is just examples of water coming into the horse and leaving the horse. Okay, so we are going to talk about how the kidneys actually do this. How do they actually get rid of stuff, and how do they actually conserve water? Okay, very, very important concept. Okay, so let's actually take a look at the anatomy of a kidney. So if you ever seen kidney beans, you know now why they're called kidney beans, because they look like little kidneys, right? So here is a setup of the kidneys in the body. All right, you can see we have 2 kidneys. Can survive with 1, but obviously 2 is better. On top is something called an adrenal gland, where it's 2 of them. This will come up. The adrenal gland is going to come up. It's going to come up later on. It's also going to come up in chapter 8. Okay, so we'll come back to that. There's your ureter. Okay, here's the bladder. You'll notice that the kidneys are attached to blood vessels, which is important because when you zoom in on a cross section of the kidney, you'll see that it's just full of blood vessels, which makes sense because I said the kidney's job is to filter out your blood. That's its job. So it's going to need a nice blood supply, right? Form and function, right? Form and function. We talked about that concept. If you know what something does, you can kind of predict its form. The kidney's job is to clean your blood, so it's going to get a nice supply of blood. Some more terminology. So the outer part of the kidney is called the cortex. This outer part, that's called the cortex. Okay, and the inner part, as we zoom in, we go towards the center. That's called the medulla. All right, so the medulla is the inner part, the cortex is the outer part. Now, what makes the kidneys work? Like, what is, because, you taught, I think in grade 10, you learn about cells and you learn about cell theory, and you learn that, when you think of like a function in your body, there's usually like specialized cells that do those functions. So, there's got to be some sort of a functional unit that works to do this for your kidneys, which is clean your blood. That functional unit is this, okay? So what you're seeing here are nephrons, all right? Now, these twisted tubes that you're seeing here, these are the units that clean your blood, all right? This is the functional unit. It's like I said, what's the functional unit of your brain? It would be like a brain cell, right? The functional unit of the kidney are these nephrons. These are what work to clean your blood. Now, it looks like it's going to be really hard to study this, so we're going to make this a little bit easier to look at, all right? But you'll notice that it looks like a twisted tube, and this is like an entrance. This looks like a snake's mouth eating something. So these are blood vessels. So we can see that there's some sort of a connection between the blood vessels and this entrance to the tubule. And I can see another one over here too. Okay, we're going to talk about that in a second. All right. So there are millions of these nephrons. These are the things that are going to clean your blood. So here is another picture. This was maybe a little bit easier to look at. You can again see the entrance, the mouth of that nephron. It looks like it's trying to eat these blood vessels. All right. This is just a much harder picture to look at, right? But it's the same idea. You can see the mouth of that nephron, all right, and which has a name, all right. It's called the Bowman's capsule. You can see it here. Right, this is that mouth of the neuron, the nephron. Okay, so here it is. There's that mouth. All right, you can see the mouth of the nephron, and there's the blood vessels. Now these are capillaries. If you remember from grade 11, capillaries are very, very thin. All right. Now the reason why they're thin is so things can diffuse through them. If you remember grade 11, you learn about like gas exchange occurring through your alveoli, which have capillaries. Nutrient absorption through your villi, which have capillaries. Capillaries are thin enough to allow substances to diffuse through them. If they're too thick, like arteries or veins, it doesn't work. So that's why the capillaries are the entrance point into the Bowman's capsule. Okay. Now, and here's another nice little picture. This is a cross section of that. Okay. So here's the idea. Think of it like the capillaries like filter paper. Okay. This unit that you're seeing here, this is a filtration unit. All right. So whatever gets through the capillaries, anything that gets out of the capillaries will enter the nephron. So either one of two things is going to happen. If I imagine that this is, let's say this is the membrane wall of my capillary, and let's say that this is the inside of the capillary, and this is the outside of the capillary, either one of two things is going to happen. Either the substance is going to go through, okay, or it's going to stay in, and because maybe it's too big and it can't go through, okay. If it goes through, it has a term, okay. The term is called the filtrate. So the filtrate is the stuff that can get through the filter paper, all right. So so in my capillaries are things that are small enough that they can get through, and I think I have it, I believe, on, yeah, here on this slide. So here are some of the things that can get through because they're small enough. So you can see glucose, bicarbonate, amino acids are small enough, but not protein. So like things like large things, like lipids and proteins, blood cells. So you're not going to be peeing out blood. That's not normal. That's not shouldn't be happening. Okay, because the red blood cells are too big to get through that membrane, the capillaries. Okay, so think of this as a filtration system. So apparently the rate at which it does this, it filters about 1.2 liters per minute of blood. All right, which apparently allows you to retrieve about 180 liters of water a day. I haven't worked out the math, but I guess that seems a little high. But anyways, this is important because 99% of that water is actually reabsorbed. If we didn't do that, then you would be literally losing about 200 liters of water a day, which you can't do. It's possible for you to lose 200 liters of water a day, right? You lose about maybe 1.5 to 2 liters of water a day because most of that material is actually reabsorbed by your kidneys. So how does it work? That's what we're going to look at. Okay, we talk about how this works. Now, to understand how this works, you just really need to remember two things: diffusion and osmosis. Yeah, if you remember those two things, this is gonna make sense to you. We're gonna make this as simple as possible. So here's my nephron. You can see my yellow tubule. It's all twisted. Here again, here's the entrance point. So whatever gets through the capillaries will go into the nephron, and either two things are gonna happen, right? It's either going to be reabsorbed or secreted. Okay, those are the opposite. So reabsorbed means it goes back in to the body. So when we reabsorb it, that means it stays in. When you secrete something, you want to get rid of it. Okay? You want to get rid of it. Most of the water is going to be reabsorbed. Most of the water is going to be reabsorbed. Most of the waste product, which is going to be urea, that's mostly what we make, is going to be secreted. Okay, not all of it, most of it. So let's take a look at how this works. Okay, again, here's the entrance point. So this is a lot easier to look at, right? So you can see here's our nephron tubule. It's what we've done is we've untwisted it because this is a lot easier to look at than something as messy as this. This is very hard to look at. So we're going to look at it this way. Now, how does it work? Okay, so again, we have the entrance point. So here's my entrance point into the Bowman's capsule. Okay, there are my capillaries in pink, and whatever gets through the capillaries, call that the filtrate. Okay, so we're going to look at really just two things: water and salt. All right, water and salt. Obviously, there's a lot more than just water and salt. There's urea and there's like other substances, but we're just going to focus on water and salt. So if you remember, the cortex is the outer part and the medulla is the inner part. All right. If I look at the slide, I'll notice that there's a something going on with the coloring, right? You'll notice that it's lighter in the cortex and it's darker in the medulla. Now, the reason for that is because there is a gradient of concentration. If I look here, you'll notice that and this refers to the concentration of solutes. I believe the unit is milliosmoles per liter. That's not really important. What's important are the numbers. You'll notice that as we go down from the cortex deep into the medulla, the concentration of solutes increases, right? Goes from 300 to 400 to 600 to 1200. All right, that's really, really important. So another way to think of it is as we go this way, the outside becomes saltier. I'll use the word saltier, okay? I'm just going to say saltier. It's easier to understand. All right, or we can say, want to be, becomes like hypertonic relative to the inside. Now, so as we go, like I said, as we go this way, the outside environment becomes saltier, right? There's more solute in it. So what happens then is, if you just remember the rules of diffusion osmosis, this is going to make sense, okay? So let's look at water and salt. They're small enough to get through, right? So you have now in the nephron tubule, you have two things. You have water and salt, right? There's your filtrate, water and salt. Now, as the filtrate is going through the nephron, it starts descending down into the medulla, right? It's going this way. This is called a descending limb. Now, as we go down, the outside environment gets saltier. So what happens then? Well, the rules of osmosis say that water goes to the hypertonic side. So as the filtrate's coming down, the water gets pulled out because the outside becomes saltier. So that pulls the water out. Now, why not the salt? Well, turns out the descending limb is permeable to water, so water can go through, but it's impermeable to salt. In other words, salt can't get through. It's stuck. Now, the consequence of this is as the water gets pulled out and the salt gets left behind, then the inside becomes more concentrated. And if you look at this slide, you see that. So we said that as the filtrate goes down, the water gets pulled out. Now, as the water gets pulled out, the inside, the filtrate becomes more concentrated, which makes sense. Because if I have a beaker of water and there's salt particles in it, now if I have less water, but I have the same number of salt particles, then the solution will be saltier. So the inside becomes more concentrated. Now, if I look as the filtrate turns through what's called the loop of Henle and goes up, I see the opposite happening. So what's going on? I see the numbers are actually decreasing as we go up. Okay, well, looks like on the other side, we have the opposite scenario. We said that as we go down, the environment is saltier, right? More solute on the outside. So the reverse is true. As we go up, the environment has less solute. So what's going on? So the other part is called the ascending limb. Now, the ascending limb is the opposite of the descending limb. It is actually impermeable to water. So in other words, water can't get out, but it's permeable to salt. So it's exactly the opposite of the descending limb. So what happens then? As the filtrate goes up, remember the filtrate became super concentrated, right? So now we just think about diffusion. Diffusion says that things want to move from a high concentration to a low concentration. So where's the high concentration? In the filtrate, in the nephron. Where's the low concentration? outside, because as we go up, the outside becomes a lower concentration. So the salt comes out. So on the descending limb side, the water comes out because the outside is saltier. Then on the ascending side, the salt comes out because as the filtrate goes up, the outside has less solute, less salt in it. So diffusion says the salt is going to move from the high concentration, which is inside the nephron, to the low concentration, which is outside the nephron. So you see how like diffusion and osmosis are really, really important concept into understanding how this works. Any questions about that? Because I'm going to show you a bit of a problem here, okay? So do you girls remember diffusion from grade 10? Did you study it? I'm sure you did. I think you should have studied when you did transport. I think it's probably drawn something like this, where diffusion is drawn like something like this, where you're going from a high concentration to a low concentration. Now, what happens in diffusion is, let's say you have A and B, okay? And on the left side, we have 100 particles. And on the right side, we have 0, okay? So 100 on the left and 0 on the right. And these particles can move back and forth, okay? They can go back and forth. Now, here's the question. Diffusion says things are going to move from a high concentration to a low concentration. So the particles are going to move to the left, to the right, okay? Now, because there's 100 on the left, there's 0 on the right. So there's a net movement, and that's a key word, by the way, is net, okay? Particles don't think. They just, they randomly move around. But because there's so many on the left, there's going to be a random movement to the right, okay? But that doesn't mean that they can't move back to the left. There's going to be a net movement to the right. But this should all balance out. Diffusion says that by the end of diffusion, things should balance out. And what you will get is, oh, my pen died. What happened there? All right. You will get like a 50-50 split. Okay, things will balance out. And, if I look at this slide, you'll notice something interesting happens at the end. It doesn't balance out. Look, if I look at what happens up here, you'll notice that there is no balance at the end. What happened there? Like how does that work? Because it should balance out so that the two sides are the same. The inside and the outside should be the same. Is that a problem? So let's see if maybe we can figure this out. I'm going to show you an animation now, okay? And this animation is going to really, really highlight what we've been talking about. So let me pull this up for you. So I came across this years ago. It talks about how it works, and you can see this now in in an animation. So that really, really, really drives the point home. Okay, so here's your nephron. It looks a little bit different, but here's the idea. Ours was, ours looked a little bit different. You can see that it's bent here, whereas in the animation, you can see that it's just straight. Oops, sorry. Where am I here? There we go. Okay. Okay, so here we go. So instead of using pink, they're going to use purple. So there's my descending limb. And there's my ascending limb. Okay. Obviously, there's more parts. We've only been talking about the descending and ascending limb part. Okay, so here is my gradient. So here they're using purple instead of pink. There are my concentrations. So it's actually milliosmoles per kilogram. I thought it was per liter, but liters and kilograms are going to be pretty much the same thing. Because 1 kilogram of water is pretty much a liter of water. So there you go. Okay, so now watch what happens. So this is the concentration of my filtrate. Okay, the stuff inside the nephron. Right now, you'll notice the numbers are increasing as we go down. So that says that water will move out. Because as we go down, water leaves because the outside is hypertonic, right? And you can see what happens. As the water leaves, the inside becomes more concentrated. You can see that right here. Now, and you'll notice it balances out, right? So that the concentration inside and outside are the same. See, inside is 1100, outside is 1100. Inside is 1300, outside is 1300. Now, as we go up, we have the reverse scenario, right? The ascending limb is permeable to the salt, but not the water. So what happens then is the filtrate is going to want, the stuff inside is going to want to diffuse out, right? You'll notice again, they balance out. So if I look at this section, right in the filtrate, there's about 1,000. So as we move up, the outside will be less concentrated. So it's going to want to come out. But you'll notice it always balances out, right? Because that's what diffusion says. Things should just balance out. Okay, but watch what happens here. It doesn't balance out. So something must be happening at this point. Okay, that's very interesting. So obviously it's not just diffusion, right? There's got to be a little bit more to this than just simple osmosis and diffusion. And the answer to that is we have actually a pump up here. All right. The pump is something called a sodium potassium pump. And the way it works is it pumps 3 sodium in one direction and 2 potassium in the other direction. So what you're seeing in the animation is sodium is being pumped out, which means then that if the sodium is being pumped out, sodium is going this way. There's my sodium, but it would be 3 sodium. Then if 3 sodium are being pumped out, 2 potassium are being pumped back in. So you can see then that if we have 3 sodium leaving, but 2 potassium returning, then there's a loss of 1 positive ion, right? So as these pumps are working, there's a net loss of positively charged ions inside the filtrate. That's why the filtrate concentration decreases because of the sodium potassium pump. This pump, by the way, we'll talk more about it when we get to the section on regulation because it can be controlled. It actually pops up again when we talk about the nervous system. This is actually a very, very important pump, okay? So that's why the concentration doesn't balance out because the sodium potassium pump actually actively pumps out more of these salt particles. That's why the concentration inside is lower than the concentration outside. Now, I'm going to wrap up. Okay, I'm going to do like 1 more minute. I'm going to show you something. If you are well hydrated, this is the end of the story. Okay, and we'll talk about this next time we meet. If you're well hydrated, that's it. Nothing else happens. You'll see nothing else happens in the animation, right? Now, if you're well hydrated, it means you've had enough water. What if you're not well hydrated though? Okay, what if you are dehydrated, which means that you have, you know, maybe it's really hot outside, maybe you're sweating a lot, and maybe you just haven't drank enough water. Then the problem is if you're dehydrated, your body wants to conserve as much water as possible. So if you look what happens when you're dehydrated, you'll notice there's a bit of a difference, right? You'll notice that there's a secondary section that is involved in the reabsorption of water. We saw it here, and now you're seeing it here. All right, you're seeing it actually in 2 locations, and that's called the collecting duct. So when you're dehydrated, the collecting duct actually helps reabsorb even more water. But that's only when you dehydrate it. So next time we meet, we're going to talk about the regulation of this because we've talked about how it works. Next time we're going to talk about how it's controlled, how it's regulated, which is a common way we do things in bio. We talk about how things work, and then we talk about how it's affected or regulated. Okay, so let me stop the record. Let me just stop the record.